everyone. This is our video of how we take glucose and turn it into fatty acids or fat rather than putting it through the Krebs cycle and getting energy out of it, or sending it off into glycogenesis or one of those other shunts, although some of those shunts from glycolysis are involved in our fatty acid synthesis, so we will look at the relationships between all of those things. So we need to think back to what we already know, which is that when we have a lot of NADH in the cell, a lot of ATP, a lot of citrate, so we're in the process of making a lot of energy. We're in the fed state, so we have, recently ingested food and we are processing it, metabolizing it, creating energy out of it. We've already got enough energy. So we start to inhibit those enzymes that help to facilitate energy creation, such as isocitrate dehydrogenase. Remember, isocitrate dehydrogenase is a key enzyme in the Krebs cycle, and if you inhibit that enzyme, it's going to slow down the Krebs cycle just after isocitrate, so just distal to isocitrate. That means that everything before isocitrate, everything proximal to isocitrate, so isocitrate itself and also citrate, are just going to build up, and we're going to get greater and greater concentrations of those. So a little bit of a positive feedback loop here if you think of citrate itself, because having lots of citrate is something that inhibits isocitrate dehydrogenase. So there's another typo, dehydrogenase. And then when you inhibit isocitrate dehydrogenase, you have more citrate building up. So what this is going to do is, instead of letting the Krebs cycle keep going on and on creating energy, it's going to be causing like a roadblock for that whole process of the Krebs cycle and the electron transport chain. It's going to be pushing all of the incoming glucose that's starting to get processed through glycolysis, etc., and it's going to be pushing it into the side pathways. So our shunts that we've talked about before, but also this new process that we haven't talked about yet, which is creating a fatty acid or fatty acid synthesis. Okay, so in the fed state, all of that citrate that is just kind of building up because we inhibited isocitrate dehydrogenase, that's going to go back out into the cytoplasm. Because remember, when we are taking acetyl-CoA, or taking our pyruvate, turning it into acetyl-CoA and reacting it with the so-called last part of the Krebs cycle, the oxaloacetate, we are doing that in the mitochondria. And when we react acetyl-CoA plus oxaloacetate to enter the Krebs cycle, we end up with citrate. Okay, but this is where we get stuck. So we end up getting all this citrate building up in the mitochondria. And that needs to go back out to the cytoplasm in this situation. So there are carriers that take the citrate out of the mitochondria, allowing it to just diffuse, because it'll be in a high concentration inside the mitochondria and lower concentration in the cytoplasm, so it will diffuse if it has a carrier that allows it to move through it, and they're called tricarboxylate carriers. Now, once we get the citrate in the cytoplasm, the citrate starts to inhibit glycolysis by inhibiting one of the key enzymes of glycolysis, which is phosphofructokinase. So when you inhibit phosphofructokinase, you are slowing down glycolysis itself. So that's even further preventing the glucose from going through glycolysis and producing more pyruvate, more acetyl-CoA, etc. Now this takes a bit of ATP, so that ATP we can use as energy, along the line, not as energy, but to create fatty acids. And it's going to be important as we start to go through fatty acid synthesis that we are going to be using ATP in order to create those fatty acids. Another thing that we need to create our fatty acids is NADPH, and that comes from our pentose phosphate shunt, which came off of glycolysis, which is going to be running a bit faster because we have inhibited the main route, glycolysis, by inhibiting phosphofructokinase. So we're going to be pushing the glucose into the shunts, such as the pentose phosphate shunt, which will give us NADPH, and then we can do our fatty acid synthesis along with the ATP and the NADPH, using both of those things. Okay. Now, also, during our fatty acid synthesis, we're going to take that citrate that is now in the cytoplasm, we're going to turn it right back into acetyl-CoA. There's also an enzyme called acetyl-CoA carboxylase, and we need all of these things for fatty acid synthesis. So we need to have ATP, we need to have NADPH, we need to turn citrate into our acetyl-CoA again. And then we need acetyl-CoA carboxylase. So all of these are necessary for us to start making our fatty acids. Now if we look at our little metabolic map over here, it's very tiny on this document camera. I have like the least zoomable document camera possible, but you don't need to see the details on this. You just need to kind of recognize some of the things that we've talked about. So here, right here, all the way into pyruvate, we have glycolysis. Okay, so this is pyruvate over here. It goes into acetyl-CoA, which goes into here. And then you should see this as, recognize this as the Krebs cycle happening in here. So the acetyl-CoA comes in, and unless we're doing the process that we're doing now, we would go through the entire Krebs cycle. There's oxaloacetate to react with the acetyl-CoA to give us citrate. But we are stopping the process there in our case because we have inhibited isocitrate dehydrogenase. So we are taking our citrate out of the process and we're turning it back into acetyl-CoA. And we're going to start doing fatty acid synthesis. Okay, now that's where our acetyl-CoA and our acetyl-CoA carboxylase is coming from. And ATP, we're in the fed state. We have a lot of ATP ready, and that's part of what inhibited us from continuing on with the Krebs cycle in the first place. But we still need our NADPH, and that's where our pentose phosphate pathway comes in. A shunt off of glycolysis, we know this. So here is the pentose phosphate pathway that's going to give us NADPH. So by inhibiting the Krebs cycle, and we can get our citrate out of there and turn it into acetyl-CoA, get some acetyl-CoA carboxylase. ATP is already there. That is what inhibited the Krebs cycle in the first place. And in turn, all of that is going to be inhibiting all the way back up through glycolysis, causing the glycolytic pathway to instead go through the pentose phosphate pathway, which gives us our NADPH. And all of these things, all of these situations combined are what allow us to start making our fatty acids. So all of that has to come together if we want to do, or not if we want to do, when we want to do fatty acid synthesis, so making fatty acids. Okay, so here are a diagram that is going into just a little more, showing it a little bit more in detail, focusing on specifically how we are using the NADPH along with the pentose phosphate pathway, and then getting our acetyl-CoA and all of that, starting to get into the specifics of making our fatty acids. So let's start out by looking at this with what we already know. Like I've said a million times, always look for what you know, what looks familiar in a metabolic map, and then just follow it step by step from there. And I know it helps if it's, let me try and get the bigger bullet points out of there and see if it is a little more clear there. Okay, so looking at this, and we see kind of a little circle situation going on over here. It's not a perfect circle like it normally is, but over here we're going to be looking at the Krebs cycle. You see pyruvate, you see acetyl-CoA, you see citrate, malate, which is part of the Krebs cycle, oxaloacetate, that's the end of the Krebs cycle, and also it says citric acid cycle, which is the Krebs cycle. So we'll just put that on there. That is the real dead giveaway right there. So, okay, we recognize the Krebs cycle, we recognize citrate and acetyl-CoA and pyruvate, oxaloacetate, all those things. Here we're kind of focusing on the citrate and the acetyl-CoA. If we go back, so follow that kind of backwards, I go, okay, well, pyruvate goes into the Krebs cycle, so we have it kind of going in a circle there. It's a little angular circle. You see glycolytic pathway, that's glycolysis, so we'll just call it glycolysis to keep life simple. There's glucose 6P, that's the beginning of glycolysis. There's glucose, there was glucose getting transported into the cell. So we've got a good amount of this that we already know and we can easily recognize. Here they're showing the pentose phosphate pathway. So over here they just kind of condensed it into blah blah blah, stuff happens and we get NADPH. So here's our NADPH coming out of there. That's necessary for making our fatty acids. ATP is coming from the Krebs cycle, obviously, and from glycolysis, but we have ATP present in the cell, just assume that. Okay, now, as we follow the rest of it, so we're looking at the kind of newer parts to us, and one we haven't talked about too much, we've mentioned it before. We've mentioned how if you have a lot of pyruvate and you don't use it in the Krebs cycle, then it turns into lactic acid and that has to go off to the liver. This is showing that little subset. We're not focusing on going off into lactic acid and going to the liver. We're focusing on all this over here, which is our fatty acid synthesis. So remember those carriers that we're going to take the citrate out of the mitochondria as it builds up, because we stop the Krebs cycle at this place here. We inhibited isocitrate dehydrogenase. Our citrate starts to build up, so we get lots and lots of citrate. Those are arrows pointing up to show a large concentration of citrate, and the citrate diffuses out into the cytoplasm. So now we have citrate in the cytoplasm. Now, there's an enzyme, and they call it ACL here. In other places, so another diagram we have later, they call it ACLY. It's the same, just, adding a Y onto the end of it. It's the same enzyme. It's called ATP citrate lyase. And this ATP citrate lyase is important for using ATP and using it on the citrate to create acetyl-CoA again. So, turning the citrate back into acetyl-CoA. And then also oxaloacetate. We know oxaloacetate. We keep seeing oxaloacetate as the end of the Krebs cycle. Also notice and remember, here's oxaloacetate going back into malate, and then we have some other reactions happening and feeding back eventually into pyruvate. But we also need to just kind of think fondly back to the malate aspartate shuttle, which we're going to revisit for a second. in a couple minutes. Just so that we can see how everything is tied together. We've seen this malate aspartate shuttle a bunch of times just because we use oxaloacetate and malate so much, and it's so much intertwined with so many of our pathways. And this is another example of, we're not just going to deplete our entire capability of doing the Krebs cycle just because we're sending the citrate out. We're going to be replenishing our oxaloacetate so that when we stop inhibiting this enzyme, we can just go right back to doing the Krebs cycle again. So oxaloacetate is just a really key molecule here for kind of switching from one pathway to the other. It's used in the Krebs cycle. It's kind of a byproduct of fatty acid synthesis, so we can go back into the Krebs cycle. Also for the malate aspartate shuttle to get hydrogens and electrons into the mitochondria for the electron transport chain. It's just involved in everything. It's tied up with pretty much everything that we've talked about so far. But anyway, I'm digressing. I'm going on to an oxaloacetate side tangent when really we want to be focusing on our citrate going into acetyl-CoA thanks to this enzyme ATP citrate lyase. Now, as we start to move from here, we're not going to think about what's happening with cholesterol over here or histone acetylation. Histone acetylation has to do with gene transcription and turning on certain specific genes. So it is an important thing, but it's just not what we're worrying about here. Here we are worrying more about how we're going to go on through the rest of this video, which is by going up into fatty acid synthesis using the molecule malonyl-CoA and an enzyme called fatty acid synthase. Notice also that the NADPH, this is where it comes in. Fatty acid synthase needs this NADPH in order to reduce or cause a reduction reaction as we start creating our new fatty acid. And that will leave us with NADP, and that can go on and get reduced again itself, essentially being recycled. So this is where the NADPH comes in. Okay, now for this slide is just looking right here. Actually, it's looking from acetyl-CoA to citrate, and then from citrate into, acetyl-CoA. So we can take acetyl-CoA, react it with oxaloacetate to form citrate. But then when we're doing fatty acid synthesis, we want to do it backwards. Okay, so it's a reversible reaction. The slide itself, or the diagram itself, shows, let's just hide that for a second. This is going to show the forward direction. So here's acetyl-CoA that came from glycolysis, or from wherever the acetyl-CoA came from, I should say. There's multiple sources of acetyl-CoA. We've seen it from glycolysis. We can get it from breaking down fats. We can get it from breaking down proteins as well. But our acetyl-CoA that so far has come from our pyruvate coming from glycolysis, it's reacting with oxaloacetate. There's that one again. That's the end of the Krebs cycle. We react the acetyl-CoA with the oxaloacetate using the enzyme citrate synthase because it's going to make citrate. And there we have citrate being produced. And if we were making energy, then we would just go on throughout the Krebs cycle. Now, because we are doing the opposite, we can do this backwards. We want to do this backwards. We want acetyl-CoA back. But now we're going to be doing that in the cytoplasm. So this is where our ATP citrate lyase, that's the enzyme that catalyzes the backwards reaction, where we take citrate and get acetyl-CoA and oxaloacetate out of it. Okay, now this slide is just focusing again on my little oxaloacetate tangent here, just because I really do want to emphasize how much everything is tied together, how intertwined all of these cycles are. And so that you really get to think about how dependent everything is on everything else inside the cell. That's one of the big take-home messages of this semester, that all of these chemical reactions are causing products that influence other chemical reactions, and they all are going to affect what what all the other chemical reactions are doing. So all of the chemical reactions are affecting each other. So we're just kind of reminding ourselves again of that malate aspartate shuttle that involves oxaloacetate, and of course aspartate, which is why it's called the malate aspartate shuttle. So if you think about, we're making fatty acids, we're in the fed state, we have all kinds of energy, we're making fatty acids, this is great, but at some point we're not going to be in the fed state anymore. We're going to start being like, okay, we need some ATP back, right? We're running low on ATP, we're using all this extra ATP, and we made a bunch of fatty acids, but now it's time to go back to making ATP. Well, when we took our citrate and turned it back into acetyl-CoA, we get our oxaloacetate there. So we're out in the cytoplasm here. We convert it into malate using any NADH present. We're essentially taking the hydrogen off of the NADH. Remember, we want that hydrogen to go into the electron transport chain in the mitochondria so we can make energy again, because we're running low on energy now. We've been making fatty acids for so long. And because of this oxaloacetate being present, we can go ahead and take that hydrogen into the mitochondria, and then we can remove it again. Now we have the hydrogen going off to the electron transport chain, making energy. And then the Krebs cycle would start to ramp up again as we start to run low on citrate. But we have like a little way of getting our energy production started again. So everything is just going from, we're just kind of switching from going from fatty acid synthesis back into running the Krebs cycle and energy production. So when we have lots of energy, we start shunting everything this way, or not everything, we start pushing some efforts this way into fatty acid synthesis. When we're low on energy, then we would slow down over here and start pushing this faster. So it's always responding to whatever situation is going on inside the cell. Okay, so just another schematic going a little bit further with our fatty acid synthesis. Again, looking for what we know, looking for what is familiar. Glucose is familiar. The pentose phosphate pathway and glycolysis, familiar. We know that glycolysis is going to give us pyruvate. We know that the pentose phosphate pathway is going to give us NADPH. Okay, we're kind of looking for both of these situations here. We're going to follow first the pyruvate, and it could go into the Krebs cycle, but we're not going to worry about the Krebs cycle right now. What we are doing is we're taking the citrate that we've created from our pyruvate, turning it into acetyl-CoA and making the citrate, not doing the Krebs cycle, but taking that citrate right back out of the mitochondria, putting it into the cytoplasm, breaking it down into acetyl-CoA. So we know that we have our ATP ATP citrate lyase enzyme here that catalyzes this step. This is oxaloacetate. This is just showing how we turn it into malate, converting into pyruvate, and kind of feeding back into this particular pathway. But we are following over this way now instead. So we're taking our NADPH. We know that we need that for our fatty acid synthesis. We know that we need our acetyl-CoA over here. It took ATP to create that. And we're going to start reacting it in the fatty acid synthesis chain of reactions. So a couple of enzymes that we have working here, acetyl-CoA carboxylase. Here it has in parentheses biotin. It uses biotin. It is not just plain old biotin, which is vitamin B7. So it uses it. So these aren't synonymous. Acetyl-CoA carboxylase is not biotin, but it has biotin as part of it. You'll also see over here pyruvate carboxylase with biotin in parentheses. That just means that it uses vitamin B7 as part of it. So it's a cofactor. Vitamin B7 would be a cofactor for both of these enzymes. Anyway, we end up with a product, malonyl-CoA. Our NADPH comes in then at this point, so it's not being added to it. It's reacting with it. We're taking the hydrogen off of it. And some more stuff happens, which we're going to get into the details of what's happening right here in a minute. But we end up with a fatty acid called palmitate. Now this is just one kind of fatty acid. It's a 16-carbon fatty acid. And we're going to look into how we can go from this fatty acid into other fatty acids as well. But first I want to point out why they have insulin just floating up here and why there's plus and plus signs right there. These little plus signs just mean that they are stimulating those enzymes, that insulin, I should say, is stimulating these enzymes. So having insulin present, such as when you are in the fed state, you release insulin in order to get glucose into cells. That insulin will help to stimulate these two enzymes, which help to facilitate this entire process, getting us to our beginning here, which is fatty acid palmitate. Okay, so let's look at our schematic of how we create our fatty acid palmitate and then how we make other fatty acids as well. Okay, so starting out from the beginning, so number one, so it's easy to find the beginning on this one. We're looking at moving citrate out of the mitochondria. This is the mitochondria in here. Here we have acetyl-CoA being turned into citrate. They have a little rudimentary Krebs cycle, citric acid cycle going on here. We're not doing that. We're stopping that. So we're taking the citrate out through the citrate shuttle. So those carboxylate carriers for our citrate carboxylate carriers are taking the citrate out of the mitochondria into the cytoplasm. Over here we are having coenzyme A. We're taking ATP. We're reacting it with the citrate. Also remember our I should put it down here actually because it's related to the ATP. ATP citrate lyase. That's the enzyme that is involved in taking our citrate and turning it into acetyl-CoA. Byproducts of that reaction, of course, oxaloacetate. Now the ATP doesn't just disappear. We just took a phosphate off of it, so it becomes ADP and sits there and waits for us to add a phosphate again at some point. Okay, so then we're going to convert the acetyl-CoA using this enzyme, acetyl-CoA carboxylase. So that should look familiar from here. So acetyl-CoA carboxylase that was stimulated by insulin, and that is going to give us our product of malonyl-CoA. So that is where we are right here. Okay. Involved with that, ATP, so more ATP getting used, so we're going to have ADP coming off of that. Biotin, remember we said the biotin was a cofactor up here for this particular enzyme, and carbon dioxide. So we're just looking for oxygens and carbons. Here they are. They are going to become incorporated into our malonyl-CoA. Now we can go to step 3, which is where we take our malonyl group and an acetyl group. Okay, so we have all of these, products kind of pre-made from steps 1 and 2. And we start with our fatty acid synthase reaction. So that's what's happening here. Remember, we need NADPH for this. And this enzyme, fatty acid synthase, which is also stimulated by insulin when you're in the fed state. And of course, we need our malonyl CoA in order for this to start happening. And we have these 4 steps here. We have condensation. I'm going to switch to a different color here so it doesn't get confusing. Condensation is when we add water to a molecule. So that's what's happening in that first step. Reduction, we know we add an electron usually in the form of hydrogen, and in this case that's what we are adding. There's that NADPH. We add the hydrogen to our newly forming fatty acid and we go back to NADP. Dehydration, we remove the water. And then we reduce again using another NADPH. So we're adding another hydrogen. Okay, so what this gives us. The first time we run that condensation reaction, so this water adding part, we're going to get 4 carbons out of that. Then everything else is going to happen 6 times, and each time it happens, it adds 2 carbons. So that adds another 12 carbons. 6 times 2 is 12. You add 4, which was the initial yield of carbon from the condensation. Then you add 12, and that gives us 16. And that is our palmitate. So we have made a nice little, fatty acid, and that is palmitate, and it has 16 carbons. Now, we're not just stuck there. We can elongate our palmitate. We can keep adding more fatty acids, more carbons. Every time we do another increment, we can add 2 more carbons and just keep kind of extending it, creating more and more fatty acids. So that's where we end up in step number 4. We're either happy with our palmitate and just, okay, we're done and that's enough, or we start elongating it. So as we start to elongate this, there's the palmitate going in. It goes through all of these, so the 4 steps: condensation, reduction, dehydration, reduction again. It's all using malonyl CoA. So, right where we started from, we just need another one of those. That's going to add 2 carbons to our palmitate as we go through these 4 reactions. There is this intermediary product that is, I don't want to say important, it's used, I should say, in this part of the reaction, the kind of initial elongation part of the reaction. And that is a CoA ester. This CoA ester, it's one fatty acid plus a phosphorylated pantothenic acid. What that means is, for an ester, anything that is an ester is when you take an OH group, so a hydroxyl group, and turn it into an alkyl group, which is just an oxygen kind of hanging out there without the hydrogen. So it would be a 2- charge on that. We don't need to worry really about that. That's not going to be something I'm going to ask you to memorize or anything like that, but just so that you understand what that is doing there, that is part of adding the elongation parts. Now also likewise, desaturation. as we continue to grow our chain, like adding the carbons, adding the carbons, adding the carbons, as we continue to grow the chain, we can have either saturation or desaturation. Saturation means each carbon has a hydrogen, or has all its bonds taken. So it's easier if I draw it. I'm not trying to say it. So I'll just put 4 here. We don't have double bonds here, or maybe we have one double bond in the middle. We know that each carbon can have 4 bonds. So this N carbon has 1, 2, 3, 4. These would all be hydrogens out here. These would be all be hydrogens. So we essentially have all the hydrogen bonds. So I'll just say all hydrogen bonded to it as possible. That's a really awkward way to say it. But all the bonds are filled up with hydrogens. If we desaturate it, we are making more double bonds between the carbons. Oops, so yeah, I put too many on there. We got to take those off. I'm counting to 4, not my strong suit. So 1, 2, 3, 4. There. Okay. So when we desaturate, let's put double bonds between all the carbons just to be extreme about it. So now this one has 2, so it can only have 2 hydrogens. This one has 1, 2, 3, 4. No more hydrogens on this one. Same with this one. This one can have 2 hydrogens. Okay, so we have a lot of double bonds between our carbons. So that's what we're thinking about when we think of saturated fats versus unsaturated fats. Polyunsaturated fats means more double bonds, like what we've just made here. Having like just a couple of double bonds would be less unsaturated, if that makes sense. And the more hydrogens you have on the molecule, the more saturated it is. Just think of it being saturated in hydrogens. No, so as we keep adding that chain, we can desaturate it with fatty acyl-CoA desaturase. So a desaturase enzyme is going to desaturate it, and that's what this example is showing. We're just growing a longer chain of a fatty acid, and we're desaturating it, so taking hydrogens off and making double bonds with our carbons. We could not do that. We could just leave it saturated, not use the desaturase enzyme and have a saturated fat, but still growing a longer chain. Okay, so to kind of recap, here is an example of creating specific other kinds of fatty acids. Anytime you see something with acid on it, it could be a fatty acid. It could be something else, but it could also say a fatty acid. This oic at the end, or an oil, that often indicates that it's going to be something that is a fatty acid. So this looks super complicated, but it's really not. We're looking at just a couple of main points. With our acetyl-CoA, our malonyl-CoA going into malonyl, so we know that stuff. We're not going to worry about the reverse part of it kind of going back in towards acetyl-CoA again and towards energy production. We're going straight into fatty acid production, and instead of using palmitate, we're just going into other more complicatedly named fatty acids, but we're just going through elongation cycles. And you can just see all the steps here where just using enzymes. When you see the ACE, that's an enzyme, so we're using specific enzymes to add specific fatty acids and to create specific fatty acids as we go through our elongation cycles. Like here, we're synthase, that means it's making something. Reductase means we're reducing something. Dehydratase, we're taking away a water. There we are, reducing it again. Thioesterase, we don't need to worry about thioesterases, but it has to do with esterification. So, taking OH and turning it into just an O with a hydrogen going elsewhere. So you can see that we have NADPH and NADP involved in this process, NADH and NAD involved as we were creating this particular, sorry, that's not the end result. Our end result is over here, this particular fatty acid. So it's just showing one very specific elongation cycle. And just remember that every time you run this elongation cycle, you're going to add 2 carbons to your fatty acid. You're going to use up 1 acetyl-CoA, 2 NADPHs, and 1 ATP. So we're essentially storing energy in these fatty acids. Now, one thing I did want to mention to you, ACP. This is the acetyl-acyl carrier protein. It catalyzes the condensation of malonyl ACP and acetyl ACP. That's this right here. So that's sort of like an offshoot. That's if you're not going to go this way into the elongation cycle, kind of like a backstop, if you will, an exit ramp to kind of not go through this elongation cycle, not create this fatty acid, but to instead just kind of go over here in a different direction. Okay, that's all we need to think about that. Okay, so what happens to our fatty acids? Palmitate and stearate. So stearate is another type of fatty acid. We saw palmitate as our main example through this video. These are esterified. Remember, we're taking an OH group and we're just, kind of stealing the hydrogen away from it. And when we esterify them, they're going to be becoming triglycerides. This happens in adipocytes and they're stored there. And then we can, break them down later if we need them through lipolysis. So remember, lipolysis is breaking down of fatty acids, where we could use them for energy. And that's usually accomplished through beta oxidation, which is another chemical process. The reverse of fatty acid synthesis. So we're instead of making the fatty acids, it would be breaking them down. But when we are storing them, we are making them, we're doing synthesis. Now, in the liver can make other kinds of triglycerides, and you've probably heard of high-density lipoproteins, which would be the so-called good cholesterol. It has more protein than fat in it. We have, or I should say, a higher proportion of protein. It's still about 50-50, a little bit less than 50-50% protein versus fat, but it's a higher proportion of protein in a high-density lipoprotein than in a low-density lipoprotein, which has much more fat proportionate to the protein. And you've heard of low-density lipoproteins as, the bad cholesterol. And these get tested every time you get a physical, your HDL versus your LDL. Now, the liver can also make something that we've probably not heard of very often, which are very low-density lipoproteins. So these are super fatty, not a lot of protein, a whole lot of fat, and they are responsible for taking excess fat to adipocytes for storage. And of course, when you eat, you're eating triglycerides. They get broken down in your gut, they're absorbed into your intestinal epithelium, which get turned into chylomicrons in there. A chylomicron is something that is where the intestinal epithelium is reconstituting the fatty acids back together a little bit after they got broken down, coating them with some proteins, and those too can be carried to your adipocytes using the lymphatic system rather than the blood so much. Well, eventually it goes into the blood. They're absorbed into the lymphatic system, which then goes into the blood and then off to the adipocytes. Okay, so our last slide, I know this is a pretty long video. I'll try to condense this end part as much as I can. This last slide is about just putting everything together, looking at 3 different places: our liver, our muscle, so skeletal muscle particularly, and then adipose, or fat cells. So we're just looking at a bigger picture rather than just right in at a cell. What we've been really looking at is fatty acid synthesis happening inside a cell. So think of, say, the liver doing fatty acid synthesis. We start with glucose, we get our pyruvate, so this is all glycolysis here. There's the Krebs cycle, there's the perfect little circle with our Krebs cycle. Do they have that right? Yeah, the TCA cycle, so Krebs cycle's written there. And then we, inhibit it because we have a lot of energy. So instead of, running the Krebs cycle, we're going to inhibit the Krebs cycle and build up citrate, which goes out into the cytoplasm again. We have our ATP citrate lyase. Remember I said it wasn't just ACL, it can be ACLY. This is the slide I was talking about. Acetyl-CoA is the result of that. Then we get malonyl-CoA. There's fatty acid synthase. Okay, making palmitate. This says elongation and desaturation. So we're, creating fatty acids. So Fa is a fatty acid. I have that written somewhere, yeah, because we have the fatty acids everywhere. There's a triglyceride that can be going up into a very low density lipoprotein, which is leaving the liver, going through the blood and getting into the adipocytes. Now, boom, triglyceride again. So we took our very low density lipoprotein, kind of broke it apart into a triglyceride. A couple of things can happen with that. It can stay there a while, or it could go right into lipolysis. So we're breaking down the fatty acid. There we have fatty acid chains. It's going to a muscle cell. We are breaking it down through beta oxidation into acetyl-CoA. Here's beta oxidation occurring here, getting us ready for our Krebs cycle again to create energy for the muscle. Now the reverse pathway, when we're taking our fatty acid and turning it into acetyl-CoA, it uses this enzyme here. I just wrote it down here. We are going to look at beta oxidation in a bit more detail later on in this unit, but just to introduce you to this enzyme, carnitine palmitoyl transferase. It's an enzyme as part of beta oxidation. So we will be looking at that later on. And then we have energy here to create, ATP for, or we have created or uses energy. We're creating ATP, I should say. We are creating ATP to use as energy for muscle contraction. So the whole process here is that we made our fatty acids in the form of a very low density lipoprotein. So just kind of elongating and elongating and elongating our palmitate, transforming it into triglycerides and eventually into very low density lipoproteins to be stored in the adipose where as triglycerides again, so broken down again. Over here we also have the adipose making fatty acids itself using extra glucose. So the fatty acid can, or the adipocyte, the fat cell, can make the fatty acid itself using fatty acid synthase for storage. Either way, whether the fatty acid came from the liver or whether the adipocyte made it itself, if we break it down back into fatty acids, do beta oxidation and get acetyl-CoA, in a muscle cell, we can use that to run the Krebs cycle and for energy. So everything tied together, but thinking from a total organism kind of place. Your liver interacting with your fat cells, interacting with your muscle cells, all dependent upon each other. And with that, that ends our glucose to fatty acid video. Our next one will have to do with oxidation, continuing on with fatty acids, being specific for fatty acids.